It's not about my work. It's about the people who are behind the work.
So what we're interested in are microRNAs, which are these small, around 22 base pair molecules that have a very important function to post-transcriptionally regulate genes. And so traditionally what everyone's looked at are these, a single sequence. But what we've shown over the last few years is that these are actually not just one, they don't constitute one molecule. So one region of the genome that produces microRNAs actually produces a whole spectrum of molecules that kind of differ based on their sequence and sort of relative to the endpoints. And what we've been able to show is that these play important roles in different diseases, cancer and other neurological diseases, Parkinson's, and importantly that these different molecules coming from the same region in the genome will regulate different sets of genes and have different properties and be associated differently between different diseases and subtypes of different diseases, and differ between men and women.
When I first joined, I didn't have that much of a science background. So what struck me the most is how, so computer science is based in math, and math is kind of like bottom up. So you start with the theorems, you start, like with the numbers, and then the theorems, and you slowly build up into like definite truths, Well, I feel biology is the opposite, where you just observe something that's very broad, and you slowly try to narrow down how it works, So for me, I think that was the hardest part, to kind of like combine two different types of thought that I feel influence what we all do and actually can make us see everything like in a different light. Computational sciences and like biology actually can make us think in different ways.
I come from more of a genetics background, and I think when you look at medicine today and the way people are treated and diagnosed, especially in the diagnosis of diseases, there's a lot of push to go into genomics and sort of sequence a gene, tumor or cancer genome, or even your own genome, and try to find different mutations that will link to disease. And there's definitely a lot of advances in that field and a lot of things that you can do with it, but that only sort of explains partial of the disease and what's going on and how to diagnose and treat your specific condition. So what we look at is more of the other parts of the genome that traditionally a lot of people thought were kind of junk and didn't really care about, but actually it will have a very big role in different diseases. And by using all this large amount of data, we could hopefully pinpoint different things that will be more specific to helping and diagnose a disease and kind of be an additional aid to sort of the traditional genomics and really improve the way we treat and diagnose diseases.
But the exciting part of that to me is that because there are so many different types of these RNAs, they might be tissue specific, and because they're very stable, they might make their way into other fluids like blood or cerebrospinal fluid, so that we can then find them there and trace them back to the source tissues to be able to diagnose disease potentially in the future, or understand how they're contributing to disease processes. So coming from the MD side of things, from the medical angle, it makes me excited because I can see a direct translation of our basic science research to the clinic in the future. Maybe not in 5 years, but in a couple decades, I think we can really do some good work in terms of diagnostics.
So in your mind, why are these RNAs important, even though we don't know what they're doing?
That's a great question. So with our new high throughput sequencing technologies, we really are able to see what exactly is going on inside the cell at a greater resolution than before. So now that we have that capacity, we can take a look at individual tissues, individual cells, and see that there are very distinct profiles of these molecules that we're studying that are consistent across tissues and disease states. So they have very, very specific signatures that would make us believe that they are important. We just have to find out what those signatures mean at this point. So we see the writing on the wall, we just have to read it now and understand it.
On what you said about translating basic research in like diagnostics, right? So the other way you could think about this is, instead of like, you know, learn about all the molecules, figure out what they do, you know about their functions, blah, blah, everything, all of these like steps that you need to take in order to end up like using them in diagnostics, you could just go on the opposite path. So basically, get blood, patients versus healthy people, see what makes a difference, see what can build like a nice classifier, and study those molecules. So why not, like, if the endpoint is diagnostics, for example, why not going from the opposite direction?
Well, I actually agree with Rogan what he said, because when I was doing my PhD, I was introduced to the world of non-coding RNA, and when I searched a little bit about it, I came to know that they are like tremendous in amount in the body, and nobody knows about their functions, and still there are many techniques, as you have mentioned, cannot capture those molecules. So first we need to find those molecules, and then definitely we need to find what they are doing in the body, and then all these diseases and all those infection studies comes up, and then we can study those molecules and their relevance to the diseases.
I had a lot of different careers before I came back to do my PhD. And the last one was I was a research technician, and I was working with chip sequencing data, and no one in the lab, it was a very small undergraduate university, and no one in the lab really knew how to analyze sequencing data. And so my boss just gave me all the data and asked me to like figure something out and find something meaningful. And so I spent like, I don't know, many 12-hour days just like going through all this data, and eventually I got really invested in it and excited about it, and I ended up going to a next generation sequencing training at the NIH. That was really fun, and I learned about what is possible, and I learned how much I didn't know. And after that, I started trying to go to grad school to basically learn how to analyze sequencing data and do computational biology, because I could see the biochemistry side of it and how useful it would be to have that in order to propel the research forward. And yeah, so I reached out to Dr. Gutzhaus, and then thankfully came here, and now I've been doing that ever since, and it's been really fun making that connection and tying those two things together.
I just didn't know your background, so it was really
Yes.
good to know, you know, what made you to join here.
Yeah, yeah, it was interesting. I didn't know either.
I think the most interesting part in this lab, in this center, is we can start from the big data, and we can actually narrow down to the molecule level. So what I'm doing now is I'm making a series of the mutation in one enzyme, and I try to see the difference in the activity of that. And in my previous experience, I did like the nucleotide examination, but here we actually look at the sub molecule, which is a modification on the nucleotide. So that is pretty exciting, and it's very tiny thing. So we can start from the big, and we can narrow down into the very tiny one. We can just actually get back to the cellular level or maybe animal level, and that is very fantastic for me.
What inspires me is this multidisciplinary interface of computer science, biology, medicine, and all the tools, techniques, molecules, and the knowledge that gets integrated into the research that I try to do, and the conclusions that I try to come to.
As a computer scientist, I feel what really makes us unique, the field of blending computer science and biology together, and things like computational biology, and why are we here today? It's really kind of a marriage of three different things going on right now. Better compute through a high-performance compute system, generation of data, lots of great data we generate here at the Kimmel Cancer Center across the street. We generate kind of genomical amounts of data. And that pieced together with wet lab expertise in advanced data analytical algorithms, we can find these novel algorithms that in novel molecules and interactions and understanding of the functions of these molecules that you've heard a lot about today. Another thing I want to add is that there's a dimension in the precision and personalized medicine dimensionality. We're all different in very unique ways. Our DNA is different and our RNA is vastly different. And what I find so special about the discovery of these molecules is we're getting insights into really what makes us unique from everybody else. Why some classes of races or sex or ethnicities have more propensities for diseases. And what we're trying to do is basically try to understand what it is that makes us different and more susceptible to these diseases, and ultimately do a better job diagnosing them.
I think computer science is very inevitable to analyze these things and develop, good tools to define the state, and at the same time, biological technique and biochemistry technique is required to understand the structure and how those expressions regulated in the cells. So combining these two things are very important to create the good things for the humans. That's what I strongly believe.
The reasons that I came here and I found this lab and I found the work that we do here very exciting is that it's a very nice combination of computational biology and building algorithms that could generalize in different types of data sets and different type of scientific questions. And we combine this with producing our own data, which is very important because you reduce any possible biases from getting different data sets around there. So I think that the way of thinking of putting this work together is very important of finding real scientific knowledge. Plus we look in molecules that haven't been evaluated or identified before. So this is the reasons. Plus when I talk with everybody here, I felt that there was a very welcoming team and everybody is super smart. So I could learn a lot through my interaction with those people. So this was the reasons that I joined. And then now currently I'm working on a data visualization course. So, and what I find exciting on that is that we can take the knowledge and the ideas that we have in the lab on how we do science and how we study things and try to train other people and think in a similar way with us. So instead of just be us, we can also make other people think in the same way, so we'll build a bigger and bigger team.